Hello everyone, how are you doing? Welcome to the Graph Data Zagreb Meetup. So some of you I know from before, some faces I see the first time, but let me give you first a bit of introduction into our community and tell you what we did so far. Okay, so we are a graph community that wanted to give a place to people from Croatia that are a part of they know something about graphs, they want to learn more about graph technologies, and we wanted to give them a place where they can speak about this. We also had a lot of fun, so our community grew over this last 4 meetups. Let's hang out more on this meetup. So what we spoke about during our 4 last meetups. So first we did a bit of Graph Technology Landscape 2022 with Dominic. Next we had a talk by Goran about detecting GitHub repository vulnerabilities with graph databases. Third talk was by me, so it was an awesome talk about Game of Thrones. And in the end, we talked about rising blockchain technologies and how they can be used with graph databases. Okay, so next we are going to hear a bit more about how you can use graph databases in pharmaceuticals. That's why we have Michaël from here. So he traveled all the way from Sweden, actually. So thank you for coming, Michaël. He will tell you, I guess, a bit more about himself, but let me tell you a story about how we actually met. So yeah, we DevRels like to hang out at Discord a lot, and we speak to a lot of people. And actually, Michaël was there. So I met him, I talked with him about graph databases in general and everything, and then we spoke a bit about amongst ourselves in Memgraph, and we noticed like, okay, so Matak also knows him, lots of people know him, so we actually noticed that our community is really strong because you can actually talk to people and you feel like you know them. But who is Michaël? So he is a graph data scientist and he works in AstraZeneca, and he is a part of Biological Insight Knowledge Graph team. And actually, he has a background in particle physics, right, and machine learning. But today, he's going to talk a bit more about this topic, so accelerating drug discovery with a biomedical knowledge graph. Actually, he's going to talk how they in AstraZeneca extract these things from biological inside knowledge graphs and distribute them to the data scientists. Did I say that all right? Yeah. Also, just a little heads up to our viewers online. Hi to everyone. So if you are watching this live, please check out the questions section and you can ask anything you want there. These questions will be answered later on in the QA sections, and then people will also have here chance to ask something, and then Michaël will answer it. Then we have a poll section. There will be one poll there, so please head over there and answer it once it's out. And if you just want to hang out, chat, and see the valuable resources that my teammate Vlasta will share, head over to the chat section. And I think that's it. I talked pretty much. And now I give the stage and give a big round of applause to Michaël. Go for it.
Okay. Thanks a lot. So, hi everyone. As Katarina said, my name is Michaël, and I work for AstraZeneca as a graph data scientist. Today I will talk to you about one of the use cases my team worked on using a biomedical knowledge graph. We are doing more than what I'm going to talk about, but it's not yet published. You know, there is a long approval process. So about AstraZeneca, so this is the fusion of a British and a Swedish company, and you know, we are producing drugs mainly in many disease areas, mainly oncology, cardiovascular, gastrointestinal disease, and a bit of neuroscience, respiratory, so like, for example, asthma. And yeah, so that's what we do in general at AstraZeneca. First, I will talk to you a bit about drug discovery. So drug discovery is a very, very long process. I think it can take up to 15 years between the moment where you say like, this gene is kind of interesting and the moment it enters the market. So first, it's very long, which means it costs a lot of money. And we are apparently in a regime of inverse Moore law. So I guess a lot of you are computer scientists or related. So you know that the Moore law, you know, you're supposed to have an increase in you know, the power of transistors. Turns out the R&D efficiency in pharma is dropping kind of low at the moment. I think it's like 2 billion per drug at the moment. So yeah, we are looking at solutions to accelerate that. So in general, humans, it will come to no surprise to you, are very complex things. And when we talk about drug discovery, we are looking at disease. And disease in general, to simplify things, are going to be caused by some dysfunction in the cell machinery into some tissues. And that, you know, at the microscopic level, you will be sick, you will have cancer, asthma, you name it. How does it look? Very close to the thing, and finally with graphs. In cells, you can imagine that the process internally can be modeled by proteins interacting with each other and passing signal to each other. So signaling that production of this other protein needs to go up, go down, and we need to communicate with other cells outside the cell. So, you know, it's a complex communication graph between proteins. And at a more macroscopic level, for the disease side of things, we have this sort of similarity graphs where we can see that diseases are related because they are caused by the same effects or they have, you know, the same symptoms. And so you end up with these two graphs. The thing is, as you can expect, we have some prior knowledge, and we know that some genes or proteins, I'm confounding the two. If you are a biologist, you are going to be, you know, mad at me, but don't be. It's in practice, it's the same thing for us. So these genes, these proteins, are sometimes associated to known disease. And we know that for quite a bunch of them. So the question of target identification is, how do we, for a new disease, figure out the good gene? Or you could also go the other way around. You have a drug, you know it acts on a gene. What is the disease that goes with it? And if you are familiar with Netflix, Facebook and all, which of course you are, it's actually a recommender system. Like, these genes are involved in the same disease. If a drug is acting on a certain set of genes, it's likely that they are going to cure the same thing, have similar side effects, etc., you name it. And we are lucky to live in an era, I don't know if we are lucky, maybe that's a big statement, but we live in an era where all these recommender systems are deployed at large scale. There is a lot of research on the topic and a lot of development. So we have a very large set of tools we can use now. So first thing is we need these graphs. This graph I showed you, and we need to integrate it into a single entity that we can then manipulate. And we must make sure that all of that has some sort of semantic meaning, it's unique definitions, etc. There are a lot of graphs in biology. So I showed you the disease network that you can see as a similarity graph. You also have like hierarchical graphs that are representing hierarchies of disease, of cell components, mechanism of actions, etc. And then you have more exogenous graph, which are protein-protein interactions that are more chemistry related, but the chemistry is in any case too hard for us to infer. So usually you measure it in the lab and you infer that things are interacting. The problem is that it's been going on for a long time, and there are a lot of projects funding such graphs, so knowledge is spread out in a lot of places. I think at the moment, in our knowledge graph, we are integrating maybe 40 data sources, or even a bit more now. And I'm only talking about the public data sources, of course. We have then our own database that we integrate later on. And that's these. Almost all of them are structured database, which means that someone has already done the job for you to say that a fact is true or not. Then there is the biggest chunk of the graph, which is actually mined into papers. And we're looking at 10 new articles every 5 minutes, every 10 minutes, every minute, which means that in general, if it takes for you more than 6 seconds to read an abstract and infer the edges described in the abstract, you are not able to catch back on literature. So our goal is to integrate all of these sources, the natural language processing outputs, into a single unified graph that we call BIKG, for Biological Insight Knowledge Graph. And this is how it looks. So we have a pipeline which is parsing all the structured databases. And the other one that is doing name entity recognition on papers, then linking the entities together according to some rules. And finally, like some proprietary data sets. And at the very end, we have this large-scale graph, which is made out of 11 million nodes. So they are the entities. So they will be cell types, genes, gene variants, drugs, compounds. All the entities that will be involved in drug discovery. In total, we have 700 million edges. So it's a very dense graph. It's actually mostly due to NLP, because a lot of edges are repeated in papers. Like you read a paper, people are always saying the same thing. Even if it's very normal or banal, they will say it because social rules say so. We can collapse these edges. So in practice, we are more working with 140 million edges in total, which is still a lot to handle. And we are often at the limit of what you can push onto a GPU, for example. How does it look in the end? How does the ecosystem work? So we are trying to do all of that for ourselves as data scientists and also for biologists. So we have different levels of exploration of the graph. The first one is what we call the explore level. So it's powered by Elasticsearch, and the goal here is really to be able to dive into all the entities. So these 11 million nodes, you search for a cancer, it must be there, and then the edges. So it's really something that focuses at the entity level, an edge, a node, what's in there. What is the context? Where did we mine it? Why was it inferred, etc. Then there is maybe something a bit more interesting for us, which is traverse the graph. So it will be pattern mining, structured queries, and it will be, for example, like, oh, can you give me all the drugs that are acting on this gene and are curing at least 2 diseases, for example? And they are questions that the biologists will ask. If I suggest them a gene, they will say, okay. They will go in the graph, this gene, is it involved in this and that disease? And that's where they are going to use either Sparkle for RGFS database, like StarDog, or Cypher for Neo4j or Memgraph or any other labeled property graph. And finally, our home as data scientists, which is more structured things. So we usually ship out the data set as a huge Parquet data set that people are downloading and playing with Python or PyTorch. And more recently, we have started to integrate it into ClickHouse to accelerate the way people are filtering the edges, etc. So that's how the full stack looks like at the moment, and it's trying to target different kind of people. So from the top, the biologists, the domain experts, to the bottom, the data scientists and machine learning engineers in the company. So now I will talk about a use case. We worked with Oncology in the past, where we use BIKG to re-rank a gene list they gave us. So the typical use case is they come to us with experimental data, a lot usually, and they want us to use BIKG to make a better decision faster, maybe with more evidence than what they would have to do themselves. And the idea is that overall we are going to throw that into some AI ranking algorithms and give them a gene list that they will screen and give their expert opinion on. So the problem we have is that we have a drug that is used for lung cancer that is targeting some kind of mutation of lung cancer. So this is this EGFR mutant non-small cell lung cancer. And the problem is that this drug is working, but there is a subpopulation of patients that is resisting to it for a reason that is not understood. So you are trying to treat people and you don't know why it doesn't work. The problem is that how resistance to drug is working is also very complicated. So oncologists decided to go to the lab and to design new cells. So they are taking model cells and they are knocking out one by one with CRISPR, which is like a way to edit the genome, the genes. And they are just either knocking them out or forcing them to be expressed. And then they treat this cell with a drug. It works, doesn't work, and we get this proxy measurement of basically the survival of cells to see if they are resisting or not to our drug. So maybe to summarize, what they try to do in the lab is to create the resistance to our drug so that we can then check how we can counteract that. So what happens is that they do like about 25,000 experiments. So it's a very lengthy and costly project. They treat it, they check manually, and they end up with 3,000 genes that could be possible sources of secondary resistance. But it's still actually too much for them to check by hand. And you need to find a way of how to mine that. So, and given that you don't have any actual labels, it has to be done a bit with external information without proper truth source. Another problem is that not only don't we have labels, but picking up a gene of choice is actually depending on things that, as machine learning engineers or data scientists, we don't really control. So it will be, is it feasible? Is it well studied? Is there a business opportunity? You have all of these discussions that are in the mind of the oncologist. So we have to integrate that. And we also have to integrate the fact that different experts will weight these things differently. So it's also part of the problem. We cannot just say maximize that, minimize that. It's not how it works. You need to be able to quickly change the way people are interacting with the ranking. So that's why we opted for to treat that problem with a multi-objective optimization problem. So essentially, each gene hypothesis, which is like saying this gene is a source of secondary resistance, is one of these dots. And we have many objectives. They are the objectives that are defined by the biologist. And we are looking for this front, like the most feasible solutions that are satisfying the criteria the biologists gave us. So maybe to give you an example, if now the objective are the tractability, which is it easy to act on that gene? Is it easy to fix? And I don't know, for example, a second objective that is so the co-occurrence in literature between the gene and the disease we are looking at. Basically, it's a way of saying, I want points that are either often mentioned in the literature, so they are well-known, well-studied, or that are easy to target. And we want a balance between those things. And this is what the Pareto front is going to show us. You know, some things where we are going then to look for the balance. What do we put into the Pareto front optimizer? It will be clinical features that are actually known by biologists, the literature support. Is it easy to drug with drugability? There are also some preclinical studies that could help. And then we just take BIKG and we ask ourselves, okay, what are the degrees of node, centralities, page rank, clustering coefficient? We run some, you know, unsupervised embeddings to have some sort of local ID of the neighborhood. And all of these numbers, we use them as feature in the Pareto ranking, and we see how they affect the ranking. We put all these features into little columns, and biologists can then say, like, oh, I want to maximize that, minimize that, and it's open source. It's in R, and yeah, it was a very nice way for oncologists to interact with the data, actually. And if they don't understand what something means, they ask us, and then they decide, like, do I want a high page rank or not? What does it mean in a cell? And essentially with that, they came with a set of criteria they liked, and they generated a list of genes they were happy with. And for us, it was a win, because if you look at the rightmost plot, which is the Shapley value of the different features, so the Shapley value being basically a measure of the importance, let's say, of the feature and the decision, you can see that the first set of features that is important for the decision are the CRISPR screens themselves, so the experimental data, the gene expression data. But the second big set of features are actually the graph-based features, which means that, you know, we could still drive a large chunk of this prediction based only on the knowledge graph. So for us, that was definitely a win. And then they checked in the lab, and turns out that, so osimertinib is the name of this drug we have that is inhibiting EGFR. And turns out that if you check how much of the cell survives as a function of the dose you treat them with, the more you emulate resistance to, the more you inhibit this gene we suggested, EZH2, the more the cell is going to resist to your drug. And like that, we have identified maybe, I think, 10, 20 possible targets that have been validated in the lab. And now it's going on. They are investigating, can we make a drug out of it? Are there any side effects, etc. So for us, it's a huge win. It's we did that with them for a year, I think. And in the end, in the lab, they can see that it's working. So it's building trust also with them on the approach. So yeah. It turns out that casting the target identification problem as a recommendation system works very well. We made the target identification faster because instead of interacting with 3,000 genes every time, we were showing them like 50 genes, and they were able to recognize them, check in the literature quickly. So it was a very good experience, I think, for them. And finally, things that were validated. So it's all good. So now, thanks to this experience, we are working with more groups in AstraZeneca, working on different set of problems with different experimental constraints, using more advanced techniques. So, for example, GNNs, the full hype wave. And we are also developing explainable AI approach and tools for them to understand why we took some decisions and not others. So, yeah, overall, it's a very interesting area to work with. And I think now it's time for questions. So the email address, if you want to work with us or have an interesting idea. There is also the GitHub of AstraZeneca, where we try to open source as much as we can. And finally, there is a career website, either if you want a permanent job or if you're a student. There is also there are also some paid internship programs. So yeah, I think now it's in Katarina's hand for the Q&A.
Thank you, Michaël. Let's first ask a few questions from the online audience. So yeah, we have one question. How do you manage the conversion between property graphs and RDF, and when do you use one model versus the other?
So it's all managed at the very end of the pipeline. Actually, we are generating the graph on Databricks with mappings to our ontology. So we have an upper level ontology to which we map everything. So we do this exercise on Databricks. We output the gigantic Parquet dataset. And once we are there, we have converters to many different formats. So we have one to project for Elastic, one for ClickHouse, one for Neo4j, one for Stardog. So overall, like, we have this single product getting out, and we convert it as we need.
Okay, so we have another question. So do you employ ontologies with the RDF graph for inferencing capabilities? And if not, do you think this would be valuable?
No, we don't, because we are just shipping it out to user at the moment. And we are not really sure reasoning will have a lot of interest there.
Thank you. So if someone from the audience also has a question, you can raise your hand and I will go to you and give you the mic. Okay, of course.
Thank you. So regarding the deployment, you mentioned Databricks, right?
Yeah.
So you have some hybrid deployment mode between on-premise and cloud, or everything is cloud-based?
No, so the build is cloud-based on Databricks with Azure. Once the build is done, it's shipped on the blob store, on the blob storage in Azure. And from there, we have pipelines fetching things, treating them. And,
Thank you. Next, did you? Are there any more questions? Yeah, you have. Okay, here you go.
Hi. How often do you recalculate everything considering new papers incoming, like stuff from NLP and so? So
we used to do it using a Microsoft Academic Graph that they stopped last year. So, you know, we had this huge dump, and every release, people were downloading everything. Now we developed our own API to do the name entity recognition on papers and download them. And we are discussing right now how to do delta processing and not reprocess everything every time, if possible, because then you have change in the graph schema. So you need to go back to square one and reprocess everything. Like, for example, last year we decided, we used to have, from a biological point of view, you have the DNA, coding for the gene, then you have a transcript, which is a piece of proteins, and then these things are put together in a single protein. We used to have the three concepts separated, but turns out that in practice, all of these things are always a three pass. It's always, you know, just increasing the radius of the graph for no reason. So we just decided to merge all of that. And the problem is that at that point, we had to go back and reprocess all the NLP stuff. But we are discussing having a more, you know, modern and granular approach to that.
Thank you. Another question?
So if I understood correctly, you're using graph properties as features for a model, like for an algorithm, for what are you using, like, in that model? So you're inputting, like, among other things, graph features. What do you use for the actual
so for the machine learning, I mean, actually it was, so we had, I think, for this example, it's only the paritory ranking, so you don't really have any learning, I think, in that example. We have other use cases where we have labels, so people know that some nodes are associated to the disease, some genes. So you can use the recall, for example, to favor them to be, I don't know, in the top 200, and then you look at the other genes that were pulled up. But it depends a bit on the use case. It could be, you know, just XGBoost, you throw features into it and try to use the non-associations as positive samples. Or it could be GCNs. We have use cases now where we use some sort of GNNs for that. So
cool. And you said, like, you validated that with, and which, maybe I missed it, which scores did you use for validation? Like,
for the validation, it's in the lab. So it's not even, it's not in silico. Like, we suggested the ranking. Biologists look at it, they say, okay, all of this is credible, let's try them out.
Okay.
And they have their own bias because, for example, the gene I showed that they picked it's because there is another cancer drug that is already targeting that. So they knew it was easy to check in the lab.
Oh, okay.
But it's actually in vivo, I mean, not in vivo, it's in vitro.
In vitro,
yeah. Validation.
Thanks.
But again, it's a bit dependent on your use case.
Just a second with the in-person. We have another online, and I'll get back to you. Okay, so how did you incorporate unstructured sources and structured sources, such as ontologies, together? And I will ask another question after that.
Yeah, basically, I mean, the NLP during the named entity recognition step, where you are looking for known biological entities, you have mappings telling you what type it is, and you just map it during the named entity recognition. So it's during the NLP analysis, you already know where are things. The relationship between the entities are actually a bit trickier, because usually we just care about co-occurrence, so they are appearing in the same abstract, in the same sentence. But in practice, we are trying to push an approach with some semantic regex, where you are able to understand what the human is saying. Like, it's just not a co-occurrence. Like, someone has said, this gene is causing this disease. And it's actually very hard, because humans tend to write in a very jargony way. We tend to like a lot of the negations. So it's kind of tricky, but we are also making progress in basically not only mapping named entities to nodes, but also mapping the relationship to a relation type we know about, and that's semantic meaning. Thank you.
And another one, can you talk about the NLP pipeline for unstructured data sources?
Yeah, as I said, we used to have a dump created by MAG and using Termite with it for named entity recognition. But now we are changing all of that. So we have this new NLP pipeline that was designed with, I think, a Korean university I forgot the name of. And we finished the development internally. But right now, we want to open source it. It's not done yet. But we have our own entity recognition pipeline right now.
We have also in-person question here.
If I understood correctly, you are working with a heterogeneous graph, right? And maybe could you tell something about whether you have ran some experiments on how that affected the model?
So, yeah, that's a bit tricky. N-1 experiments where you would remove, for example, types, or we've talked a lot about it. Actually, the heterogeneous nature of the graph is not really used right now at the machine learning step. It's more upstream, people filtering the graph. It's really that. It's people who don't like this provenance. I don't trust this lab. I don't trust this type of experiment. I only want genes that are particularly expressed in, I don't know, kidney, or this organ or another. So in terms of models, it's quite rare to use the actual type of the relationship. We've had some use case where people use the metapath, the concept of metapath, but it stops there. Right now we are working with some GNNs that are aware of the heterogeneous relationship. But overall, it's important for the human to explore the graph, but not necessarily from a machine learning point of view.
Thank you. Any more questions? Yeah. Oh, many questions.
Yeah, hi. Do you stream data from the Elasticsearch to Neo4j or something else?
No. Elasticsearch is so we would like eventually to move to a more streamlined way of releasing things with deltas and such. Right now, we have one graph release every 2 months. So we just like one big indexing in Elasticsearch.
Like a dump or something like that.
Yeah, we have a dump, a projection meant for Elasticsearch, and then we index it. It takes like maybe a day or two, and then it's deployed.
Why are you using ClickHouse?
Yes, it's more recent, yeah.
Mm-hmm. Okay, and second question, how many people... Oh, sorry. How many people works with you in your team on this particular project?
So BIKG is, I think, 14 people. In these 14, there are a few vacancies that are, we have open position, and we are trying to grow the team to serve more use case and, reinforce the technological side of things.
Okay, thanks.
Okay, we had one more here.
Hello. You have mentioned using a XGBoost.
Yeah.
As I understand it, it's mainly used on tabular data.
Yeah.
So really like to know how did you square it with a graph data set?
That's the thing. In practice, we are treating a tabular data set. That is a list of genes that you were interested in. We try. That's a difficult question in the sense that you would like to use the graph structure itself, but in practice, it's not very practical. I mean, it's very hard to work with graph at scale, and it's much faster to just featureize the entities, like fetching page rank in this and that projection of the graph, featureize your nodes, and then run a classical algorithm. First, because it's easier to run, and in a week you can have results, which is important for the people you are talking to, and also because you can explain them much more easily. Also, that's really practical reasons. So,
thank you.
Okay, we have one here.
So are you processing only abstracts or whole text?
So right now in the graph, it's still using the NLP from the old dump, so it's only abstracts. But now the new API is using abstract and full body for, I think, for bioarchive. And then it depends a bit, there are licensing like fees and such for Elsevier, Wiley and such. I think we have worked out agreements with them, and we will be able to process the full body.
So what's your suggestion for writing better abstracts?
I don't know, but that would be a very interesting analysis to make, actually. Like,
yeah, because I'm a reviewer for lots of biomedical papers, so there should be there should be some suggestions for that.
Yeah, I think, we have started to discuss in the team, like, first trying to characterize abstracts. From an NLP point of view, when we are inferring the edges, essentially each abstract, or even a paper, it's telling a story. It should be told in the graph, this gene causes this disease, and we tested it in this and that. It's not necessarily the case. Sometimes it has multiple disconnected components. So, overall, I've been publishing in the past in my previous life. The abstract, sadly, it's not necessarily meant for science. It's a very social exercise, and people are going to use a lot of buzzwords, acronyms, and for NLP, it's a gigantic pain, because EGFR, this gene, it can actually mean 3 or 4 different acronyms depending on which subfield you're looking at.
So do you also take into account statistically negative results, because there's always bias and forced to publish only statistically significant?
Yeah. So when we are training embeddings, for example, for the graph, it's a long-standing question, like, why do we have to sample negatives? Some things are known to be wrong. There are obvious wrong facts, things that have been documented, but there is none in the literature. As you said, like, there is so much incentive in saying that your work is a success, that mining actual negative edges is super hard. And there used to be a journal, actually, that was publishing only negative results. And this guy closed, because we were saying, like, let's just use them. So now we are looking more at solutions that people, like, for example, in KTH are developing and such, where you have an implicit negative sampling, but still we are in an open world. So essentially in the graph, every edge that doesn't appear is supposed to be false. And you just hope that statistics and neighborhood will make things better.
And also my last question, how AstraZeneca decides what will be open access or proprietary?
So as far as we're concerned, code, everything can be open source. Essentially, like, we are trying, like, we have, for example, released an ontology mapper recently. We are going to release an ontology parser. There is chemical X. We are trying to release packages on our end. The dataset itself, I think we've been discussing opening it up for a year. The problem is that you need to purge the proprietary data, and then we are paying some license to access some datasets. And we need to clear with the seller that it's okay to distribute, and then you have a lot of lawyers involved, compliance, etc. So one day there will be an open BIKG, I hope, but But essentially, management let us do what we want as long as we don't endanger the assets of the company. So.
Okay, thank you.
Thank you. So we have another online question. Let me just jump there. Okay, so have you considered pushing for nano publications to make your life easier?
Pushing for?
Nano publications.
No, we haven't. The problem is that publishing is, as long as we keep it in our corner of science and it's machine learning and all, that's perfect. The moment you are touching biology and there is a potential drug at the end, it becomes a very lengthy process, you know, and you are targeting bigger journals, so it's a bit more complicated.
Does anyone else here have a question in person? Is that all? Okay. Thank you, Michaël. Give him a big round of applause.
Thank you. Thank you, everyone.
Thank you. Okay, so thank you all for your questions. You'll be able to talk with Michaël also later on. And in the end, Dominic, okay. I just want to share with you our resources that our team created. So our MAGE team actually did a little demo with Memgraph and Chemical X. So it's a deep learning library for drug-drug interaction. And they created a whole demo and a blog post that is explaining it in detail. So I will send you these links, of course, in the Meetup group. You can click on it and check it out. If you want to check it out now, you can head over to the Memgraph blog and check the latest blog posts. And also we have the creators here, Josip and Antonio, so you can also ask them more about this. Our team also created this Memgraph Playground lesson. So they gather the data from the biomedical knowledge, network of biomedical knowledge, and there you can explore different kinds of diseases such as lung cancer, and meanwhile also exercise your cipher knowledge. In the end, if you really love C++ and like dream about it at nights, then join our strong core team. They always love new graph and C++ enthusiasts. And yeah, as I said, we have a strong community. Join us if you also want to be a part of our community and maybe speak one day about your projects, share them with us. And yeah, we like GitHub stars, so throw us one. And thank you. I think that's all. There will be burgers outside, like always, beers, drinks, everything. Cheers, enjoy. I hope you like this. And thank you again, Michaël, for coming to our meetup.